Hi, my name is Nora Shekem binti Zulkifli.
Nuclear transplantation is a method in which in this chapter is referred as somatic cell nucleus transfer (SCNT), in which in the nucleus of the somatic cell, which is donor cell, is transferred into an unclotted egg or oocyte, which is the recipient. However, other examples of the nuclear transplantation involve non-somatic cells such as sperm, fetal cell, embryonic stem cell, and germ cell are using the term interspecies SCNT, which means when donor and recipient cell come from different species, family, order, or classes. Nuclear transplantation has allowed experimental embryologists to manipulate the development of an organism and to study the potential of the nucleus to direct development. Nuclear transplantation, as it was first called, was later referred to as somatic nuclear transfer of cloning.
I am Aina Azin binti Abdul Nasir. Here, I want to talk about who can do or undergo the nuclear transplantation. We can undergo the nuclear transplantation in animals. Before that, do you know who is Dolly? Dolly is the first successful in a mammal's clone from an adult somatic cell. Dolly is a female sheep that was born in 1996 at Scotland. Sadly, Dolly was put to sleep at age of 6 because she has a progressive lung disease and severe arthritis. After cloning was successfully demonstrated through the production of Dolly, many other large mammals were cloned, including pigs, deer, and horses. For human nuclear transplantation using somatic cells, there is no laboratory has succeeded. This is because for safety and efficiency of the procedure and the intensity extent of ethical objection.
Hi, I'm Nur Akila binti Zahari.
Vesdila first wrote about nuclear transplantation in 1895, speculating that if one were to replace an egg nucleus with another egg nucleus, full development will occur. Later in 1938, Hans Berman suggested an experiment whereby, using technologies not yet available to him, one could remove the nucleus of an egg and replace it with a different nucleus extracted from a developed cell. Thomas King and Robert Briggs were the first to perform experimental nuclear transplantation. The technique was soon after used by John Gurdon and eventually led to the first clone of a mammal, Dolly the sheep, by Lance Willman in 1996. During this nuclear transfer is carried out, the addition of genetic material or production transgenic animal is achieved by the injection of a required gene into pronucleus of a zygote. Although this technique has been applied successfully in a number of species, including mice, rabbits, pigs, sheep, goats, and cattle. In contrast, the production of offspring from a single cell or clone population offers significant advantages. Genetic modification can be performed in culture, and the modified cells selected prior to animal production.
Hi, I'm Izzati binti Abdul Aziz. What are the type of artificial cloning? There are 3 types of artificial cloning: gene cloning, reproductive cloning, and therapeutic cloning. They are all different types of cloning. Gene cloning involves the creation of copies of gene or segments of DNA. Gene cloning, also known as a molecular cloning, refers to the process of isolating a DNA sequence of interest for the purpose of making multiple copies of it. Reproductive cloning is a process that involves the transfer of cloned embryo into an artificial uterus. This procedure produces copies of whole animals. Therapeutic cloning is a technique used to extract stem cells from cloned embryos. This procedure works by removing the cell from the embryos and then transferring them to a uterus. It is creating tissue to replace a diseased tissue. Gene cloning is a different process from reproductive and therapeutic cloning that involve transferring DNA. Reproductive and therapeutic cloning is the same technique, but it is done for different purposes.
Hi, I'm Miminor Azira binti Zamri. As we know, nuclear transplantation is a form of cloning and self-evidently known as somatic cell nuclear transfer. Now I will explain how this nuclear transplantation is done in sheep cloning. First of all, the scientists will remove the somatic cells from the mammary gland cells of a donor animal and let them be cultured in vitro. Next, an unfertilized egg cell is obtained from an ovum donor and the nucleus will be sucked out, leaving the cytoplasm itself. The somatic cell and egg cell will then given an electrical pulse to fuse the cells together. The cells that are fused are now called an early embryo. It is then implanted into a surrogate mother which has the same breed as the donor sheep. Lastly, the cells are allowed to divide repeatedly, naturally until they become a complete embryo, and finally, the cloned sheep was born.
Hi everyone, I am Lina Nadira binti Abdullah.
Why nuclear transplantation is used? This is because nuclear transplantation can preserve endangered species. For example, in China, panda cells are being kept on reserve if this species number be threatened by extinction. The second reason is because the nuclear transplantation can give us the opportunity to produce the most desirable traits because we use the nucleus of the desired animal. The third reason is because we can reproduce the healthiest animal, thus minimizing the use of antibiotics, growth hormone, and other chemicals. The last reason is we could advance scientific discoveries in medical science because could potentially take the technique discovered in this field to produce new tissue or organ as needed. Even though there are many benefits in nuclear transplantation, but nuclear transplantation also have many disadvantages, which is the sign of cloning is unsuccessful a majority of the time, which is 95% of cloning attempts end in failure. This is because of higher risk of birth defect, impairment, or susceptibility to illness. The next disadvantage is cloning animals reduce the genetic diversity of that species. It is also expensive, and nuclear transplantation also can raise ethical issues on human cloning.
Thank you.